Good day. Our lecture for biochemistry would be catabolism of proteins and of amino acid nitrogens. Objectives for this lecture would be to explain the metabolic fate of amino acids that are catabolized in the body, identify the major enzyme systems responsible for degrading damaged or unneeded proteins, such as ubiquitin, transaminases, and glutamate dehydrogenase. Give the role of the stomach, pancreas, and small intestine in protein degradation. Give the role of the urea cycle in nitrogen metabolism. Identify the sources of nitrogen atoms in urea. And give the symptoms of hyperammonemia and their causes. Why is it important to know these things? Proteins and amino acid catabolism describes how the nitrogen of amino acids is converted to urea. In normal adults, nitrogen intake should match nitrogen excretion. When disease sets in, this balance is greatly affected. Positive nitrogen balance is an excess of ingested over excreted nitrogen accompanying growth and pregnancy, whereas negative nitrogen balance, output exceeds intake, may follow surgery, advanced cancer, kwashiorkor, or marasmus. While ammonia, derived mainly from the alpha-amino nitrogen of amino acids, is highly toxic, tissues convert ammonia to the amide nitrogen of non-toxic glutamine. Subsequent deamination of glutamine in the liver releases ammonia, which is then converted to non-toxic urea. If liver function is compromised, as in cirrhosis or hepatitis, elevated blood ammonia levels generate clinical signs and symptoms. This is a summary of how it is metabolized in our body. We ingest proteins through our diet. This is degraded by pepsin and converted to our polypeptides and amino acids, and then converted to your oligopeptides and amino acids, further broken down to your aminopeptidases and di- and tripeptidases. Then utilize amino acids either by the small intestine and then to the liver. Again, this is a model of your dietary protein metabolized as your trypsin becomes your chymotrypsin and eventually your elastase and would eventually become your carboxypeptidase A and B as it goes along the small intestine. Protein turnover occurs in all forms of life. Each day, humans turn over 1 to 2% of their total body protein, principally muscle protein. High rates of protein degradation occur in tissues undergoing structural rearrangement, such as uterine tissue during pregnancy, tadpole tail tissue during metamorphosis, or skeletal muscle in starvation. Of the liberated amino acids, approximately 75% are reutilized. The excess nitrogen forms urea. Since excess amino acids are not stored, those not immediately incorporated into new protein are rapidly degraded. Proteases and peptidases degrade proteins to amino acids. The susceptibility of a protein to degradation is expressed as its half-life, T to the 1/2. Half-lives of liver proteins range from under 30 minutes to over 150 hours. Typical housekeeping enzymes have T to the 1/2 values of over 400 Rs. By contrast, many key regulatory enzymes have a T to the 1/2 of 0.5 to 2 hours. PEST sequences, regions that are rich in proline, glutamate, serine, and threonine, target some proteins for rapid degradation. The resulting peptides are then degraded to amino acids by endopeptidases that cleave internal bonds. Aminopeptidases and carboxypeptidases remove amino acids sequentially from the amino acid carboxyl terminals, respectively. Extracellular membrane-associated and long-lived intracellular proteins are degraded in lysosomes by ATP-independent processes. By contrast, degradation of abnormal and other short-lived proteins occurs in the cytosol and requires ATP and ubiquitin. So what is ubiquitin? Ubiquitin is a small protein that targets many intracellular proteins for degradation. The primary structure of ubiquitin is highly conserved. Only 3 of 76 residues differ between yeast and human ubiquitin. Several molecules of ubiquitin are attached by non-peptide bonds formed between the carboxyl terminal of ubiquitin and the amino acids of lysyl residues in the target protein. This is how ubiquitin is processed in the body. It is acted on by 3 enzymes: an activating enzyme, a ligase, and a transferase. The carboxylic terminal of ubiquitin first forms a thioester. The coupled hydrolysis by pyrophosphatase ensures that the reaction will proceed readily. A thioester exchange reaction transfers the activated ubiquitin to enzyme 2. Then enzyme 3 then catalyzes the transfer of ubiquitin to the amino group of a lysyl residue of the target protein. Additional rounds of ubiquitination result in subsequent polyubiquitination. The residue present at its amino terminal affects whether a protein is ubiquitinated. Amino terminal met or serine retards, whereas aspargine or arginine accelerates ubiquitination. Degradation occurs in a multicatalytic complex of proteases known as the proteasome. This is the glucose-alanine cycle, wherein the glucose is utilized in the muscle, transformed to pyruvate, then eventually alanine, and this alanine is transported to pyruvate and then converted back to glucose. The rate of hepatic gluconeogenesis from alanine is far higher than from all other amino acids. The capacity of the liver for gluconeogenesis from alanine does not reach saturation until the alanine concentration reaches 20 to 30 times its physiologic level. Following a protein-rich meal, the splanchnic tissues release amino acids while the peripheral muscles extract amino acids. In both instances, predominantly branched-chain amino acids. Branched-chain amino acids serve a special role in nitrogen metabolism, both in the fasting state when they provide the brain with an energy source and after feeding when they are extracted predominantly by muscles. How are dietary proteins digested? Proteins are hydrolyzed to amino acids for absorption to make them smaller. Proteolytic enzymes responsible for degrading proteins are in the stomach, pancreas, and small intestine. In the stomach, gastric juices such as hydrochloric acid and pepsin are present. The hydrochloric acid kills bacteria, and pepsin is from pepsinogen, which is activated by your hydrochloric acid. In your pancreas, pancreatic proteases further cleave polypeptides initially digested by the stomach. Enteropeptidase from the intestine activates pancreatic enzymes such as trypsinogen. The small intestine contains aminopeptidase that cleaves oligopeptides to produce free amino acids. Free amino acids and dipeptides are absorbed by the intestinal epithelium. Dipeptides are further hydrolyzed in the cytosol before being released to the portal system. This is the post-absorptive state from your stomach, pancreas, and small intestine. Again, your proteins are utilized by your kidneys and then excreted by your liver through urea and glucose, with your muscles, brain, and your liver utilizing your glucose energy. In animals, the aqueous environment of telosteian fish, which are ammonotelic, meaning excretes ammonia, compels them to excrete water continuously, facilitating excretion of highly toxic ammonia. On the other hand, birds which must conserve water and maintain low weight are uricotelic and excrete uric acid as semi-solid guano. Many land animals, including humans, are ureotelic and excrete non-toxic water-soluble urea. High blood urea levels in renal disease are a consequence of impaired renal function. And urea biosynthesis occurs in 4 stages. The first is transamination. Second is your oxidative deamination of glutamate. The third is ammonia transport. And 4 is reactions of the urea cycle. The first process, transamination, interconverts pairs of alpha amino acids and alpha keto acids. All the protein amino acids except lysine, threonine, proline, and hydroxyproline participate in transamination. This reaction is readily reversible, and aminotransferases also function in amino acid biosynthesis. This is your structural representation of your transamination between your alpha amino acids and your keto acids. The coenzyme pyridoxal phosphate is present at the catalytic site of aminotransferases and of many other enzymes that act on amino acids. During transamination, bound PLP serves as a carrier of amino groups. Following removal of alpha amino nitrogen by transamination, the remaining carbon skeleton is degraded. Alanine pyruvate aminotransferase and glutamate alpha ketoglutarate aminotransferase are 2 enzymes that catalyze the transfer of amino groups to pyruvate, forming alanine, or to alpha ketoglutarate, which forms glutamate, as shown in the reactions below. Each aminotransferase is specific for one pair of substrates, but non-specific for the other pair. Since alanine is also a substrate for glutamate aminotransferase, all the amino nitrogen from amino acids that undergo transamination can be concentrated in glutamate. This is important because L-glutamate is the only amino acid that undergoes oxidative deamination of an appreciative rate in mammalian tissues. The formation of ammonia from alpha amino groups thus occurs mainly via the alpha amino nitrogen of L-glutamate. N-Glutamate dehydrogenase occupies a central position in nitrogen metabolism. Thus, the transfer of amino nitrogen to alpha-ketoglutarate forms your L-glutamate. The release of this nitrogen as ammonia is then catalyzed by hepatic L-glutamate dehydrogenase, which can use either a NAD class or NADP class. Conversion of your alpha-amino nitrogen to ammonia by the concerted action of glutamate aminotransferase and GDH is often termed trans-deamination. Liver GDH activity is allosterically inhibited by ATP, GTP, and NADH, and activated by ADP. The reaction catalyzed by GDH is freely reversible and functions also in amino acid biosynthesis. And listed in this diagram would be the fates of the amino group of various amino. L-amino acid oxidases of liver and kidney convert amino acids to an alpha amino acid that decomposes to an alpha keto acid with release of ammonium ion. The reduced flavin is reoxidized by molecular oxygen, forming hydrogen peroxide, which then is split to oxygen and water by catalase. The ammonia produced by enteric bacteria and absorbed into portal venous blood, and the ammonia produced by tissues and rapidly removed from circulation by the liver and converted to urea. Thus, only 10 to 20 grams per deciliter normally should be present in peripheral blood. This is essential since ammonia is toxic to the central nervous system. Should portal blood bypass the liver, systemic blood ammonia levels may rise to toxic levels, and this occurs in severely impaired hepatic function. Ammonia intoxication include tremor, slurred speech, blurred vision, coma, and ultimately death. Ammonia may be toxic to the brain in part because it reacts with alpha-ketoglutarate to form glutamate. The resulting depleted levels of alpha-ketoglutarate then impair function of the tricarboxylic acid cycle in neurons. Formation of glutamine is catalyzed by mitochondrial glutamine synthase. Since amide bond synthesis is coupled to the hydrolysis of ATP to ADP, the reaction strongly favors glutamate synthesis. One function of glutamine is to sequester ammonia in a non-toxic form. Hydrolytic release of the amide nitrogen of glutamine as ammonia, catalyzed by glutaminase, strongly favors glutamate formation. The concerted action of glutamine synthase and glutaminase thus catalyzes the interconversion of free ammonium ion and glutamine. An analogous reaction is catalyzed by L-asparaginase. Excretion into urine of ammonia produced by renal tubular cells facilitates cation conservation and regulation of acid-base balance. Ammonia production from intracellular renal amino acids, especially glutamine, increases in metabolic acidosis and decreases in metabolic alkalosis. This is the urea cycle. It's a series of reactions that produces urea from ammonia, converting it to the non-toxic form. This was the first metabolic cycle discovered. In mammals, the urea cycle takes place only in the liver. Functions of the urea cycle include organisms that cannot easily and quickly remove ammonia usually have to convert it to some other substance like urea or uric acid, which is much less toxic. Insufficiency of the urea cycle occurs in some genetic disorders and in liver failure. The result of liver failure is accumulation of nitrogenous waste, mainly ammonia, which would eventually lead to hepatic encephalopathy, a neuropsychiatric abnormality. The urea cycle consists of 5 reactions. 2 occurs in the mitochondria and 3 occurs in the cytosol. The cycle converts 2 amino groups, one from ammonia and one from asparagine, and a carbon atom from bicarbonate. The relatively non-toxic excretion product, urea, at the cost of 4 high-energy phosphate bonds, 3 ATP hydrolyzed to 2 ADP, and 1 AMP. Ornithine is the carrier of this carbon and nitrogen. Urea is the major end product of nitrogen catabolism in humans. Synthesis of 1 mole of urea would require 3 mole of ATP plus 1 mole each of ammonium and the alpha amino nitrogen of aspartate. No N-Acetylglutamate functions as an enzyme activator. So the initiator is Carbamoyl phosphate synthase 1. Condensation of your carbon dioxide, ammonia, and ATP to form carbamoyl phosphate is catalyzed by mitochondrial carbamoyl phosphate synthase 1. This is the rate-limiting enzyme of the urea cycle. This enzyme is active only in the presence of its allosteric activator, N-acetylglutamate, which enhances the affinity of the synthase for ATP. Formation of carbamoyl phosphate requires 2 moles of ATP. So the initiating enzyme is carbamoyl phosphate synthase 1 in the mitochondria, requiring 2 moles of ATP. Defects in the enzyme would be responsible for the relatively rare metabolic disease termed hyperammonemia type 1. The second reaction is carbamoyl phosphate plus ornithine forming citrulline. L-ornithine transcarbamoylase catalyzes transfer of the carbamoyl group of your carbamoyl phosphate to ornithine, forming citrulline and orthophosphate. The reaction occurs in the mitochondrial matrix. So we are still in the mitochondria. So we are at the second reaction with ornithine and citrulline. So the star of the second reaction is the ornithine transporter. Hyperornithinemia, hyperammonemia, and homocitrullinuria syndrome, or your Triple H syndrome, results from mutation of the ornithine 1 gene that encodes the mitochondrial membrane ornithine transporter. The failure to import cytosolic ornithine into the mitochondrial matrix renders the urea cycle inoperable. The X chromosome link deficiency termed hyperammonemia type 2 reflects a defect in ornithine transcarbamylase. The mothers also exhibit hyperammonemia and an aversion to high-protein foods. Levels of glutamine are elevated in blood, cerebrospinal fluid, and urine, probably as a result of enhanced glutamine synthesis in response to elevated levels of tissue ammonia. Argininosuccinate synthase links aspartate and citrulline via the amino group of aspartate and provides the second nitrogen of urea. The reaction requires ATP and subsequent displacement of AMP by aspartate forms citrulline. So we are at our third reaction outside the mitochondria, already at the cytosol. So cleavage Cleavage of argininosuccinate, catalyzed by argininosuccinase, proceeds with retention of nitrogen in arginine and release of the aspartate skeleton as fumarate. Addition of water to fumarate forms L-malate and subsequent NAD plus dependent oxidation of malate forms oxaloacetate. These two reactions are analogous to reactions of the citric acid cycle but are catalyzed by cytosolic fumarase and malate dehydrogenase. Transamination of oxaloacetate by glutamate aminotransferase then reforms aspartate. So our subsequent reactions occur in the cytosol. Arginine is produced from argininosuccinate. The ASL gene is located on chromosome 7. When deficient results in argininosuccinate accompanied by elevated levels of argininosuccinate in blood, CSF, and urine. Argininosuccinate is associated with friable, tufted hair, which is your trichorexis nodosa. Hydrolytic cleavage of the guanidino group of arginine catalyzed by liver arginase releases urea. The other product, ornithine, re-enters liver mitochondria for additional rounds of urea synthesis. Ornithine and lysine are potent inhibitors of arginase, competitive with arginine. Arginine also serves as the precursor of the potent muscle relaxant nitric oxide in a calcium 2+ dependent reaction catalyzed by nitrogen oxide synthase. Hyperargininemia is an autosomal recessive defect in the gene for arginase. Unlike other urea cycle disorders, the first symptoms of hyperargininemia typically do not appear until age 2 to 4 years. Blood and CSF levels of arginine are elevated. The activity of carbamoyl phosphate synthase 1 is determined by N-acetylglutamate. N-acetylglutamate level is dictated by its rate of synthesis from acetyl-CoA and glutamate and its rate of hydrolysis to acetate and glutamate. These reactions are catalyzed by N-acetylglutamate synthase and N-acetylglutamate hydrolase. N-acetylglutamate is essential for carbamoyl phosphate synthase 1 activity. The gene encodes N-acetylglutamate synthase, which catalyzes the condensation of acetyl-CoA with glutamate. Defects in the NAGS gene result in severe hyperammonemia, which may respond to administered N-acetylglutamate. Again, this is your major changes in diet can increase the concentrations of individual urea cycle enzymes 10 to 20-fold. Starvation elevates enzyme levels to cope with the increased production of ammonia. So all defects in urea synthesis would eventually result in ammonia intoxication. The effects are most severe when the metabolic block occurs on reactions 1 and 2 in the mitochondria. For if citrulline can be synthesized, some ammonia has already been removed by being covalently linked to an organic metabolite. Clinical symptoms common to all urea cycle disorders include vomiting, avoidance of high-protein foods, intermittent ataxia, irritability, lethargy, and severe mental retardation. Significant improvement and minimization of brain damage accompanies a low-protein diet ingested as frequent small meals to avoid sudden increases in blood ammonia levels. Treatment could be feeding high doses of benzoic and phenylacetic acid, which are excreted in the urine as conjugation products with glycine and glutamine, respectively. Gene therapy also offers promise to correct defects in urea biosynthesis. So it rectifies defects in the enzymes of the urea cycle is an area of active investigation. The preliminary results are encouraging, for example, in animal models using an adenoviral vector to treat citrullinemia. So this is a table form of your urea cycle reactions. The first two occurring in your mitochondria. The last three would be your cytosol. Okay. So for this lecture, we have obtained the following objectives. Thank you for listening to this biochemistry lecture. Please do subscribe to my channel for more lectures on Biochemistry 1. Thank you.